Hey everyone, Dr. D here, and in this video we are starting a new lecture series on Biology 2420. This is microbiology for non-science majors. Typically students who want to go into nursing or dental hygiene or the like. The textbook we'll be covering is this one here, Microbiology, A Systems Approach by Cohen. This is the 7th edition. That's the textbook we'll be focusing on. But, I used to teach from the Bauman book, and, these concepts really don't change much. Regardless of what textbook you're using, you'll probably be just fine. My cats, Gizmo and Wicket, also help me with this lecture series. You'll see them pop in and out with tips and tricks. You could tell them apart because Wicket has the spot on his nose. I constantly tease him. I always ask him, who put Sharpie on your nose? So that's Wicket, and the other one, Gizmo, accused once of being the cattest looking cat ever. They help me out by popping in and out of the videos as well. So I hope me, Gizmo, and Wicket can help you to learn microbiology this semester. So let's go ahead and get started with Chapter 1 of this lecture series.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, let's go ahead and get started with Chapter 1, the main themes of microbiology. I want you to know, first of all, what is microbiology? It's the study of microorganisms and infectious agents. These are the things that they refer to as germs, right? When they say don't spread germs, try not to sneeze in people's face, try not to cough without covering your mouth. You're spreading germs, things that are communicable, these types of things like bacteria, viruses. That's what you're here to study. That's the focus of microbiology. It's a little bit more broad than just bacteria and viruses, though. So we're going to get into what all these terms mean. And the first part of the lecture, I'm going to tell you what all are germs, because it's not just bacteria and viruses. There's more to it. And then we need to know, what groups they belong to, the different germs belong to, what the different germs look like, how they're spread, how they grow. That's basically what you're here to understand is what do they mean by germs and how do they spread? And the fact that it's micro means that they are too small to be seen with the eye, right? So your eyeball, believe it or not, has a limit in terms of how small object you can see. And that limit is right here. It's 0.1 to 0.2 millimeters. You guys know what a millimeter is, right? A millimeter. Well, think of a tenth of a millimeter or 0.2 of a millimeter. That is how small you can see with your eyeball. It's called the resolution limit of the naked eye, the human eye. Anything smaller than that, you're not going to see. And so anything smaller than milli, we're talking about the micro world, the micrometer world, because this is something interesting to know. 1 millimeter is 1,000 micrometers. Okay, knowing that means that the eyeball can see down to 100 micrometers, right? Because 0.1 millimeters equals 100 micrometers, okay? Knowing that, you should know that microorganisms are smaller than 0.1 millimeter. They are smaller than 100 micrometers. They are in the micro realm, right? That's why they're known as microorganisms, because they're micrometers big. And if something's in the micrometer range in terms of its size, you can't see it with the naked eye. And this is why we need microscopes, right? So we use microscopes to study the microorganisms in microbiology. So let's get into it a little bit deeper. First of all, what are the major groups of microorganisms and infectious agents? And what's the difference between a microorganism and an infectious agent? Let me explain that real quick. when you hear organism, like microorganism, you should think of a living thing, okay? Something that's made up of at least one cell. And do you remember the 3 domains of life? Hopefully you learned this in biology 1406, general bio, but there are 3 domains of life: bacteria, archaea, and the eukarya, which envelop some of these other characters here. Those are the 3 domains of life. And if you are an organism, if you are a living thing, you belong to one of those 3 domains of life: bacteria, archaea, or eukarya. You and I belong to the domain eukarya because our cells are eukaryotic. We are made up of eukaryotic cells. So when we're talking about the major groups of microorganisms, we're talking bacteria, which are prokaryotic. They belong to the domain bacteria. We're talking about things like E. coli or salmonella. All bacteria are microorganisms. All bacteria are single-cell organisms that are prokaryotic. And remember the difference between prokaryotic cells and eukaryotic cells? We're going to get into this in a couple slides, but remember, prokaryotic cells lack a true nucleus. Sometimes they're called akaryotic cells because A means without. And eukaryotic cells have a true nucleus. They both have DNA, by the way. Did you know that bacteria has a chromosome? Archaea has chromosome. And eukaryotes have multiple chromosomes. But in eukaryotes, those chromosomes are enveloped in a membrane, right, called the nuclear envelope. So because the DNA is inside of a membrane, that's why eukaryotes have a nucleus. Bacteria and archaea, their DNA is not inside of a membrane. Their DNA is just floating around in the cytoplasm. So their DNA is not considered inside of a nucleus. I'll tell you what their DNA is called in a little bit, but foreshadowing, it's called a nucleoid. Now, I digress. Let's get back to what are the major groups of microorganisms. All of the domain bacteria are microorganisms. This includes E. coli, salmonella, some of the other characters you've heard about. Then there's a domain archaea, which are also made up of, prokaryotic cells. They are all single cell organisms. In fact, under a microscope, archaea and bacteria look about the same, right? They look the same. They're both prokaryotic cells. Okay, archaea, prokaryotes, bacteria, prokaryotes, all are microorganisms. You know why? Because you can't see them without the assistance of a microscope. They are micrometers in size. Now, what about eukaryotes? The rest of these 3 here, these 3 are eukaryotic microorganisms. Now, this is interesting because eukaryotes, like you and I, the domain eukarya, it includes some large animals like you and me, humans, but it also includes microorganisms, single-cell organisms as well. So some of these organisms include some of the fungi, right? These include molds as well as yeasts. So those are microorganisms studied by microbiologists. There are a group of eukaryotes called protists. You guys know what protists are? These are typically, not always, but typically single-cell eukaryotes. These include the protozoa. You may have heard of, for instance, amoeba or paramecium or giardia, right? Those are types of protozoa. And then algae as well. These are photosynthetic protists. Okay, so the protozoa and the algae are types of protists, which are, typically single-cell eukaryotes, and these are studied by microbiologists. And lastly, this is the strange one. Microbiologists study helminths. Helminths is another term for worms. Now, look what it says here. Not technically a microorganism. See, when you might be thinking, but Dr. D, why do microbiologists study helminths? Because helminths are worms, right? And I see a worm, it's pretty big. I see when it rains, the earthworms come out and I can definitely see them. They're not micro at all. But keep in mind, worms cause human infection, and it's typically the eggs that get into your body that give you a worm infection. See, if you eat undercooked pork or you drink from a contaminated river, right? You might ingest the microscopic eggs, and that is how you obtain the worm. The worm itself might be macro, but the life cycle stage in which you ingested it or it infected you is typically micro. And this is why, the combination of the fact that helminths cause human infection, and it's typically the eggs that give you the initial infection, that's why helminths are studied by microorganisms, okay? So these first 5 bullet points here, these are organisms, right? And do you know why they're organisms, even though they're microorganisms? Because they are all made up of at least one cell. Do you agree? So helminths, protists, fungi, archaea, bacteria, these are all organisms. They are alive. To be alive, you have to belong to one of the 3 domains of life, right? So if you belong to bacteria, you belong to archaea, you belong to eukarya, well, then you belong to a domain of life and you are alive, right? Now, but is that all that can be spread from person to person and make you sick? Or are there other tiny things that can be spread that are not technically organisms? Have you guys heard of viruses or prions? These are actually smaller and more simple than cells. They are not cells at all, actually. A virus is essentially not much more than either DNA or RNA surrounded by some protein, right? And a prion is actually only made up of a protein. So these are not organisms, are they? Your textbook kind of refers to them as an organism, but I think that's kind of, a misnomer. I would call these not organisms, not alive. So what do we call them? We call them non-cellular, because they're not cells, non-cellular small infectious agents. So I want you to remember that viruses and prions are not alive. They are not organisms, but they can be spread person to person. They are very small, very, very small, and they can cause disease. Oh, look who's here to help me. Which one are you? You're the one with the Sharpie. It's Wicket. Wicket's come to say hi and help me. Wicket's always invited to help me instruct. Wicket's going to paw at the screen, I'm sure, in a minute. So hopefully this slide makes sense to you guys. When you're talking about viruses, they are so much smaller than cells. They are not alive. They are small infectious agents, and prions as well. We're going to learn a lot more about all of these. Don't worry. We're going to learn about viruses in detail, prions in detail. But so far, I just want to give you a big perspective on what is studied by microbiologists. And these are the types of organisms and infectious agents that are. Now, what's interesting about studying microbes is that it can be both easy and difficult at the same time. Studying microbes is easy because they reproduce rapidly. We're talking about every 20 to 30 minutes, these bacteria can double. So they can form huge populations in the blink of an eye, well, within a few hours, right? But the problem is they can't be seen directly, right? Remember, they're in the micro realm. They're smaller than the 0.1 millimeter resolution limit of the eyeball. So we need to use indirect methods to see them. We need to use microscopes. Does that make sense? Now, what do microbiologists study? They study everything about the microbial world. They study the cell structure, function, growth and physiology, genetics, taxonomy, evolutionary history, interactions with living and non-living environment. Basically, you name it. If there is a niche in the microbial world, there are microbiologists studying that niche. And here are some branches of microbiology that you might consider going into. There's medical microbiology, dealing with microbes that could cause disease in humans or animals or even plants. Public health, you've heard of the CDC or the WHO, the WHO, that monitor and control the spread of diseases. There's immunologists who are trying to understand how your immune system battles these intruders, when these microorganisms get into your body. There's industrial microbiology, which safeguards our food and our water. They use biotechnology, so they can use these microorganisms to make us products that we can use. Isn't that cool? There's, like I said, so many different facets to microbiology. There's so many ways that we can use these microorganisms to our advantage. Agricultural microbiology, figuring out how these microbes can help us with crops and crop growth. Environmental microbiology, figuring out how we can use either these microorganisms or their products in order to help us with the environment. For instance, if there's an oil spill, can we use microorganisms to break down that oil and help us to remediate that lake or that environment? Okay, so microbiology is a vast field, and hopefully you got a sense of what types of organisms and agents we study and all the different areas of study. And if you're in 2420, likely you're going towards medical microbiology, you're going towards nursing. So you have to understand how these microorganisms can cause disease, right? So if you're a nurse or a dental hygienist, you have to understand, how to protect yourself against these microorganisms, how to protect your patient, how to prevent the spread of these germs from patient to patient, how to keep the area sterile. All that stuff is covered in microbiology. So it's a really interesting and fun topic of study. It's very, very, very important for you to understand microbiology because, you need to know how these bugs spread in order to prevent their spread. And if you're in this class, you're most likely going to be dealing with that in your career. Now, these microorganisms, namely the prokaryotes, were the first to form on the planet billions of years ago. And it took more than 1 billion years for eukaryotic cells to appear. Now, what was interesting was the Earth's atmosphere was originally anoxic. This means that there was no oxygen in the atmosphere on Earth. And it was those early prokaryotic photosynthesizers that produced oxygen in the environment. Isn't that interesting? Again, remember, there are two main types of cells that organisms consist of: eukaryotic cells and prokaryotic cells, sometimes referred to as akaryotic cells, A meaning the absence of. Eukaryotes have a true nucleus. This means that their DNA is inside of a membrane. Bacteria and archaea, they are prokaryotes. That means that they don't have a true nucleus. That means their DNA is just floating around inside of the cell's cytoplasm. The DNA is not inside of a membrane. And remember, the term for this is a nucleoid. Again, I want you to remember there are 3 major domains of life: eukarya, bacteria, and archaea. All bacteria and archaea are microorganisms, are prokaryotic, while some eukarya are microorganisms. Obviously, you and I are eukarya, right? We belong to the domain eukarya, and we are made up of eukaryotic cells, but we're not microorganisms, obviously. We're multicellular creatures. But there are so many microorganisms that are eukarya, and those are the ones studied under the umbrella of microbiology. Now, one thing about bacteria that you should understand as a microbiology student is that bacteria are pretty much ubiquitous. Ubiquitous is a fancy term for everywhere. Bacteria are everywhere. They are ubiquitous. They can be found everywhere on the planet. We're talking deep in the Earth's crust, at the polar ice caps, the oceans, on your body, on the bodies of plants and animals. Pretty much every surface on the planet has bacteria that you can find. And so bacteria are ubiquitous. In fact, did you know this fun fact? There are more bacteria and other microorganisms on your body right now than you have your own number of cells. Isn't that interesting? And let me tell you something really interesting. The bacteria on your body, these bacteria exist on your surfaces, okay? Not in your tissues. Now, you might be confused because you might be thinking, but I heard there's lots of bacteria in my intestines, in my colon. That's inside of me, isn't it, Dr. D? Well, think about this. Humans are kind of like a tube, right? We're kind of like an elongated tube. And so if you think about a human, we have a mouth, right? And that mouth proceeds to the throat and then down the esophagus to the stomach, down to the intestines, and then the large intestine, the colon, and the anus, right? All of that is actually your surface, isn't it? For instance, if I touch the inside of my cheek, that's still my surface. That's not inside of me, is it? Inside of me would be like in my blood, in my heart, in my tissues, my organs, right? That's what I mean by inside. So think about this. The microorganisms that mainly reside on your body, they're on you. They're not in you, okay? I know it's hard to get to wrap your brain around, but think about this. If there's bacteria in your mouth, that's still on your tissues. That's not inside of your body. That's still on the surface of your body, right? And so the bacteria inside of your throat, they're still on your surfaces, right? The bacteria in your stomach, they're still not inside of you. They're on the surface. They're on the surface of your stomach. Now, if the bacteria were to bore through your stomach and go into your blood, then that would be more technically inside of you. Does that make sense? So when biologists or microbiologists state that there's more bacteria on your body than you have your own number of cells, they're not talking about in your blood, okay? If you were to draw your blood, like sterilely draw blood, and look inside your blood, you shouldn't find microorganisms in your blood. You shouldn't find bacteria in your blood because that's the role of your immune system. Your immune system defeats those and destroys those. In fact, that's a sign of a systemic infection. If you have blood that's contaminated with bacteria, that means you have a bad infection that you're fighting off, right? So your organs should be devoid of any organisms, any bacteria, microorganisms. It's your surfaces, including the surface of your mouth, the surface of your throat, esophagus, stomach, intestines, large intestine, colon, anus. This is where you find lots of bacteria, but technically, again, this is your surface. Even though it's on the inside of your body, it's not in your tissues. So think about that, okay? Inside of your body, there shouldn't be any microorganisms. It's also important to remember this concept of evolution, that life evolved from a common ancestor, and this was the result of descent with modifications, traits passed on generation to generation as they adapt to their environment and having changes in gene frequency due to selective forces, natural selection. And again, as I mentioned earlier, some of the earliest microorganisms were photosynthesizers, and they are the ones that contributed to the oxygen in the environment. In fact, even up to this day, it's the microorganisms. We're talking about photosynthetic bacteria called cyanobacteria and algae, like green algae and diatoms. These are the creatures that make up more than 70% of the Earth's photosynthesis, and they contribute 70% of the oxygen on Earth. A lot of times students, when you think of what's providing the oxygen for us to breathe, what's providing the photosynthesis on the planet, students will often say, oh, it's plants, it's trees, right? But that's the minority, actually, of the photosynthesis occurring. It's really this algae, it's the diatoms, it's the photosynthetic bacteria. Those are the ones that are pulling most of the weight. Isn't that interesting? Bacteria and fungi, they also play a very important role in recycling organic matter in the environment. We're talking about decomposition when an animal or a plant dies. It's these decomposers like fungi, bacteria that come in and break down that dead organic matter, recycling those nutrients so that the circle of life can continue. And as I mentioned earlier, these microorganisms, they are so useful. As humans, we have been using these microorganisms to benefit us in so many different ways. Everything from production of various food and beverages like wine, beer, bread, cheese production, right? Antibiotics and such. We use them for many, many, many of the products that they can produce for us. In the field of biotechnology, we are manipulating these microorganisms so that they can make industrial products for us, to make products that benefit humanity. And with recombinant DNA technology, we're able to introduce genes into microorganisms so that they can produce things for us and to help us to provide different enzymes for medicine, different types of proteins such as insulin for diabetes, many different organisms, these organisms that are genetically modified, GMOs, genetically modified organisms, benefiting humanity. And again, I mentioned earlier bioremediation, using these microbes or the enzymes they produce to help clean up toxic pollutants, including oil spills and such. Look how broad the scope of microbiology is today. But of course, that's not to say that all of it is wonderful and roses. A lot of these microorganisms and small infectious agents are pathogens. They cause disease. A pathogen is any agent or organism such as a virus, bacterium, fungus, protozoan, helminth, any one of these types of entities that cause disease. And there's nearly 2,000 different microbes or agents that cause disease that we know about today. For instance, if we look at the top causes of death by all diseases in the US, some of them are caused by microorganisms or infectious agents. For instance, COVID-19, which is a viral communicable disease. Communicable means spread from person to person. Also influenza or pneumonia, another one that cracked into the top causes of disease. Now, if we look at the top causes of death worldwide, we have even more examples of microorganisms or infectious agents. See, here you see influenza and pneumonia, COVID-19. We also see diarrheal diseases, which are often caused by microorganisms as well. So here you can see these terms communicable versus non-communicable. Communicable means being able to spread from person to person or from animal to person, right? These are caused by microorganisms or infectious agents versus non-communicable diseases. When you think of non-communicable disease, think of, heart attack or stroke, right? This is a disease, but it's not caused by the spread of a germ, for instance. So if we look at this, 30% of worldwide deaths are caused by infectious diseases, these communicable diseases. And since 1990 till more recently, 2015, We've had decreases in some infectious diseases such as measles, diarrheal diseases, but we've seen an increase in non-infectious diseases such as diabetes. In the US, we see a very low incidence of number of deaths per 100,000 people of the infectious origin versus non-infection. If we look in the world, it's still more pronounced. Infectious disease still accounts for many more deaths. However, you can see it's the non-infectious diseases that are predominant. And that's a good thing because it shows how far we've come in the field of microbiology to provide antibiotics, to provide vaccines, to help prevent the spread of disease from communicable diseases. Now, a major example of a communicable disease that kills about 500,000 people every year is malaria, which is spread by mosquitoes and caused by a protozoan. This is again, a mosquito spread disease, and the microorganism gets into your blood and causes serious illness. And a lot of bed nets are used to prevent these mosquito bites. And recently, a vaccine has been produced against malaria, which is very, very promising and very exciting. But getting that vaccine to susceptible populations is a challenge. But again, an example of a communicable disease that causes many deaths is malaria. And we're going to talk more about the pathogen that causes malaria, which is a type of protozoan called Plasmodium. We're going to talk about that later on in another chapter. And at any point in time, there are always new, or also known as emerging diseases coming around, like, the ones we've been struggling with the last few years, COVID-19, as well as older diseases that are coming back around or re-emerging, such as, Ebola or AIDS, for instance. These are all diseases that have cycles, new diseases popping up, old diseases coming back, right? There's so many different diseases and their dynamics vary. And so this is why it's so important to have establishments such as the CDC and WHO constantly monitoring this so that we can get them under wraps. There's also some good news. Polio, leprosy, parasitic worm diseases have largely been eradicated based on our knowledge of microbiology and our efforts to limit the spread of these diseases. There are even diseases that were once considered non-infectious, but now we know are caused by communicable microorganisms or infectious agents. For example, did you know that ulcers, gastric ulcers, like if you have a stomach ulcer, it used to be thought that that was caused by spicy food or stress or lack of sleep or, who knows what, or not eating enough. Well, now we know that it's mainly caused by a bacteria known as Helicobacter pylori. Isn't that interesting? There's also a link between certain, but not all, cancers and bacteria and viruses, mainly viruses. Viruses can cause certain cancers, and also a link between Coxsackie virus with diabetes as well as schizophrenia. MS, multiple sclerosis, OCD, coronary artery disease, obesity, all of these have had links with chronic microbial infections as well. Isn't that interesting? So every time we think we know it all, there's something that comes around the corner to show us that there might be more to the story. Isn't that interesting? All of these were once thought to be caused by non-communicable forces, but now at least to some extent, these are shown to be linked with microorganisms and infectious agents. And remember, as I mentioned earlier, you should not have microorganisms inside of your blood, inside of your tissues, right? And that's because your immune system does such a good job clearing infections. But if people have weakened immune systems, these people are subject to infections. This is because their immune system is not robust enough to clear those infections as they are in healthy people. And another topic of study with regard to microorganisms and pathogens is this recent, deluge of drug-resistant microbes. These are bacteria that used to be easily treated with antibiotics, which are now becoming more and more resistant to antibiotics, such as penicillin. So we need to be monitoring these as well. So there are many new problems arising in microbiology, and there are many old microbes that we thought we had a hold of that are now becoming more and more resistant, that are giving us challenges today. Let me quiz you and see if you can beat my cat Wicket to the answer. What are the 3 domains of life? That's right, Wicket. Archaea, eukarya, and bacteria. Those are the 3 domains of life. Very good, very good. You got it. And remember, if you do not belong to one of these 3 domains of life, then you're not a living organism. And let me ask you this. Let me see if you can beat Wicket again. Do viruses belong to any of these 3 domains of life? That's right again, Wicket. Wicket's always right, by the way. I've only seen him be wrong once before in biology 1406, but he's usually right. He's a star student. Yes, viruses are not considered living entities by most scientists, and that's because viruses do not belong to archaea, eukarya, or bacteria. Viruses aren't even made up of cells. They're made up of DNA or RNA and a protein coat, right? So they are very, very simple. The majority of microorganisms are single-cell creatures, such as all of the bacteria are single-cell, archaea are single-cell, and eukaryotes have example of single-cell organisms, right? Eukaryotes, remember, eukaryotes have fungi that are single-cellular. Not all fungi, but many fungi are single-cellular, especially the yeasts. Eukaryotes have those protists. Remember the protists, which include the algae as well as the protozoa, right? Those are single-cell organisms as well. And then remember we mentioned earlier some microorganisms are multicellular, such as some of the fungi and the helminths, the worms. Also, you should realize that bacterial and archaeal cells are much smaller than eukaryotic cells. And that makes sense, right? They don't have a nucleus. They don't possess membrane-bound organelles. Your textbook says that they lack organelles, but your textbook's a little bit misleading there. The bacteria and the archaea lack membrane-bound organelles. They lack organelles that have their own membrane. Do you guys remember the term organelles? Let me explain that real quick. Organelles are functional parts of the cell. Just like organs are functional parts of you, right? You have organs such as the heart, the brain, the liver, right? Cells have organelles, which are functional parts of the cell. So organelles include things like the nucleus, the mitochondria, the chloroplast, right? You've heard of those things hopefully before. Well, bacteria and archaea, they not only lack the nucleus organelle, but they lack other membrane-bound organelles that have membranes around them, right? Such as mitochondria, chloroplasts, endoplasmic reticulum, Golgi apparatus, lysosomes, right? So bacteria and archaea don't have any of those structures inside. And if you don't have any of those structures, those bulky structures, you're going to be a lot smaller and a lot less advanced, right? Does that make sense? So it's no wonder that bacteria and archaeal cells are 10 times smaller than eukaryotic cells. Now, again, I mentioned that bacteria and archaea, they still have organelles. For instance, they have ribosomes, right, which are organelles. Ribosomes are an organelle, but they don't have their own membrane. Does that make sense? That the ribosome doesn't have its own membrane, so it's not known as a membrane-bound organelle. So again, the best way to phrase it is bacteria and archaea lack membrane-bound organelles, such as the nucleus, the mitochondria, etc., etc. So let me tell you something. Again, do you remember the resolution limit of the eye? Can you beat Wicket? What was the resolution limit of the eye? That's right, Wicket. It was 0.1 millimeters, also known as 100 micrometers, right? 100 micrometers. Well, let me give you some other sizes here to think about. If the smallest thing I can see with my eyes is 100 micrometers, well, the average eukaryotic cell is about 50 micrometers across, in diameter. And that means that the average prokaryotic cell, the average bacteria or archaea, is only about 2 to 5 micrometers across. Does that make sense? So the bacterial and archaeal cells are tiny, tiny, tiny compared to eukaryotic cells. But eukaryotic cells are already, much smaller than what we can see with our eye. So the reason I bring that up is because when you're in microbiology and you head to lab, and you bust out your microscopes and you're all excited to look under the microscope, you're going to see those eukaryotic cells no problem because they're fairly large microorganisms, right? Those eukaryotic cells are about 50 micrometers, and your microscopes have no problem resolving those cells. But did you know that with the light microscope, like the one in your student lab, even at the maximum setting on that microscope, like the highest magnification possible, 1000x on your student microscope, those bacteria still are tiny. I mean, you really have to squint to see most bacteria, even at 1000x with oil and everything all set up at the maximum setting. Those bacteria are still tiny. Like you have to squint, and you can pretty much only tell if they're round or long, like capsule shaped or round shaped. That's all you can tell about them. That's how small we're talking. These bacteria and these archaea are tiny, tiny, tiny little guys. And let me ask you this. Do you think viruses, if you could barely see bacteria with a microscope and a bacteria is a whole cell, do you think you can see viruses with our microscopes in the lab? Remember, viruses are much, much, much simpler than cells. They're just DNA or RNA with a protein coat. Let me see if you can beat Wicket. That's right. Again, Wicket. No, you can't see them. You can't see viruses with our student microscopes. So you should realize, like, for instance, if you were trying to do a study with SARS-CoV-2, the virus that causes COVID, you couldn't look at that virus with our student microscopes with what's called a light microscope because they are so small, you're not going to see them. You need a fancy electron microscope to see a viral particle, right? A virion, it's called. So I hope I gave you an idea of just how big the size difference is between eukaryotes, prokaryotes, and viruses. Again, eukaryotic cells contain membrane-bound organelles like the nucleus, mitochondria, and chloroplast. Bacterial and archaeal cells, they only have non-membrane-bound organelles such as the ribosome or the nucleoid. And again, remember, viruses and prions, these are not alive. These are known as acellular, that means non-cellular, infectious agents. These are not organisms. They are not alive. Viruses are composed of either a DNA genome or an RNA genome, and it's wrapped up in a protein coat, right? So it's basically DNA or RNA with a protein coat, and that's not a living entity, okay? And then prions as well. Prions, these are just proteins that are misfolded, that cause disease, that are communicable, that can cause disease. Again, your 6 types of microorganisms. You've got worms or helminths, fungi, protozoa, bacterium, viruses, and prions. And remember, technically, viruses and prions, these are not organisms. These are agents. So now let me give you some backstory, some history to microbiology. Who were the fathers of microbiology? Who were the people that really brought the field to life and helped us in so many ways? It's one thing I want you to know is that people back then, they knew that food spoiled, but they didn't know why. They knew that people became ill, but they didn't know why. For instance, when the Black Plague hit Europe and wiped out most of Europe, when smallpox hit, people didn't know what was causing it. It was very terrifying time. They knew it was some kind of transmissible matter, but they didn't know what. They thought it was like they thought it was winds or forces, or that your body fluids. They thought you had 4 different body fluids, and if they got out of whack, you got diseased. They thought all kinds of things, right? But they didn't know what caused disease. They didn't know what caused food spoilage. In fact, back then, they believed in a concept known as spontaneous generation, that life could come from nothing, right? So for example, if you leave a steak on the counter, next thing you know, little maggots grow out of the steak, right? So back then they thought, well, the maggots just formed out of, the steak. Life came from nothing, right? And so this was the concept of spontaneous generation. It was really a confusing time before let's say the mid 1600s and on, right? That's when the field of microbiology really hit the scene, and it started answering some of these questions, like why does food spoil? What causes illness? What causes these other illnesses like Black Plague, smallpox? Why do maggots seem to grow out of meat, right? What causes fermentation? People didn't know any of these concepts. Why is it important to wash your hands? Why is it important to sterilize surgical instruments? Why is it important to, cover your mouth when you sneeze? Why is it important to refrigerate your food, right? None of these things were understood until starting the mid 1600s with some pioneers, which we're going to talk about now. So one of the fathers of microbiology, Louis Pasteur, in the mid to late 1800s, he was a rock star, an early pioneer in the field of microbiology. He showed so many groundbreaking discoveries. Let me just list a few before I show you how he disproved spontaneous generation. He was the one who definitively disproved the concept of spontaneous generation. He contributed to the principles of vaccination, along with another scientist, Jenner. He demystified fermentation. Vintners, winemakers hired him in order to discover why wine was, so wonky. Sometimes you would make a good batch of wine. Sometimes the wine would be not a very good batch of wine. Well, he discovered what causes fermentation. He discovered that fermentation is a result of yeast doing an anaerobic process to produce alcohol. Isn't that interesting? He was the one who discovered how fermentation really works so that winemakers could make consistent batches of wine and beer, etc., right? Isn't that interesting? And he proposed the germ theory of disease, that diseases are caused by germs like bacteria, and they're spread from person to person. Isn't that interesting? So look at this. I mean, any one of these would earn someone a Nobel Prize, right? But he discovered 4. And these are not, it goes on and on, by the way. I can include to this list many other things. Like, have you ever heard of the concept of pasteurization, like pasteurized milk? That was him. It's named after him because he discovered how to pasteurize, how to treat milk so that the taste is retained, but the microorganisms are for the most part killed off, right? I mean, like I said, I could go on and on about Louis Pasteur's discoveries, but the four most notable are right here. Let's talk a little bit more about the first one, how Louis Pasteur disproved spontaneous generation. So it was known back in the 1800s that broth would go bad, right? Think of chicken broth or beef broth, right? If you make beef broth and leave it out, what happens? It goes bad, right? It becomes rancid. So what people thought at the time was that the broth goes bad because organisms spontaneously form in the broth, right? And the reason they thought this was because even if you boiled the broth like this, even if you boil the broth, if you leave it out, well, then it would go bad on its own, right? So they thought boiling the broth should kill anything in the broth. And if it kills anything in the broth, then leaving it out shouldn't allow it to go bad. But broth would go bad. Well, what Pasteur thought was that perhaps something was landing in the broth from the air, like dust, let's say dust or spores or debris from the air, was landing in the broth and causing the broth to go bad. So in order to show this, what he did was his famous swan neck experiment. This is known as Mendel's famous swan neck experiment. He would take the broth, he would boil it for quite a period of time inside of the flask. The microbes are getting destroyed in the flask, and so he would kill the microbes in the broth. Then, this was the genius part of his swan neck flask experiment, he would draw the neck of the flask into a swan neck structure, and then he would make sure that the end of the neck was open so air could go in and air can come out. There's air exchange. So, because if you seal the flask, if no microorganisms form, the critics would say it's because, well, of course, no spontaneous generation occurs. Well, because air is not getting into the broth. So what Louis Pasteur did was he boiled the broth, killed all the microorganisms, then he drew the neck out as a swan neck. So air could get in, air can get out, and then he waited for the spontaneous generation to occur. So let's see what he saw. Let's go over here. So this is the boiled broth with his swan neck that's open, and look what it says. The broth was free of live cells. It remained sterile for months, right? It would remain sterile. If he left the neck, the swan neck intact, it wouldn't spoil. It would remain fresh, right? Even at room temperature, the broth would remain fresh. And do you know what he noticed? He noticed that any dust, any debris, any spores would get stuck in the swan neck. So the dust and debris, which carries bacteria, it would get stuck in this neck and it would never make its way into the broth to spoil the broth. However, if Pasteur broke the neck, right, shortly after breaking the neck, the broth would spoil, showing that dust and debris getting into the broth delivered the microorganisms to the broth. The broth didn't spontaneously generate the microorganisms. The microorganisms were deposited into the broth from the air, right? And the swan neck being intact prevented that from happening. Isn't that neat? So by doing this elegant, simple experiment, Louis Pasteur disproved the concept of spontaneous generation. And again, this happened in the mid-1800s. At this point, finally, people accepted that life didn't just grow out of nothing, right? That, cells come from pre-existing cells. Isn't that interesting? Another important development in the history of microbiology, of course, was the development of the microscope. The development of the microscope really began in the 1600s. And two people whose names are synonymous with early microscopes are Robert Hooke, who made early lenses that were able to resolve fungi and other small organisms, as well as Antonie Van Leeuwenhoek, who was a haberdasher. If you don't know what a haberdasher is, it's a person who sells linens and mainly men's outfits, a men's outfitter. So think of like Men's Warehouse or, gentlemen's suits and top hats and such. So Antonie Van Leeuwenhoek, he was into fine linens and fine cloth, and he would make lenses to look at those cloth at close, look at the thread count and look at the fine silks and stuff that he was selling. Well, he got really good at fashioning lenses to look at more and more objects up close until he was able to form this structure here. He formed this apparatus. This is known as his famous pinhole microscope. Van Leeuwenhoek formed this microscope. He put a lens that he fashioned, a lens in this little hole, and then he would put his specimen, whatever he's trying to study, on the tip of this pin. So he would take muddy water, or he would even take stool sample, and he would put it on the tip of this pin, and he could see tiny microorganisms. This lens magnified up to 300x. Isn't that interesting? So what you would do is hold it kind of at this end, hold it at this end, and hold it up to your eye and look through the pinhole of the pinhole microscope. Isn't that neat? So by that, he made drawings of what he called animalcules, right? So almost like molecular animals is what he called them. Now we know them as microorganisms. Isn't that interesting? So it was really, again, Robert Hooke and Antonie Van Leeuwenhoek in the 1600s who were the first to fashion these crude, early, simple microscopes and see these microorganisms such as fungi and protozoa and such under the microscope. Modern microscopes are quite more advanced. They have refined lenses. They have a structure known as the condenser, which we'll talk about. It condenses the light, a finer focusing device. So we have fine focusing, coarse focusing knobs. We have built-in light sources. We have two lenses in a compound light microscope. So our microscopes are quite a bit more advanced. They're stronger. They can magnify up to 1,000x, right? So we can see small bacterium under the microscope. And what about the development of early medical microbiology? Early experiments showed that microbes are everywhere. Remember, they are ubiquitous. Even the air has dust, which is full of microbes. There's dust floating around in the air. Bacteria piggyback on that dust. There are fungal spores in the air. So anywhere one of these spores lands can become a mold infection. Okay, so remember, I said bacteria and microorganisms are ubiquitous. They're all over the planet. And it really wasn't until some of these pioneers in the field of medicine started to figure out that not only are microbes everywhere, but they are contributing to disease. That's when patient outcomes became so much better because, back in the day, before we understood the role of germs and disease, doctors wouldn't wash their hands. They wouldn't sterilize or even disinfect their surgical equipment, right? Think about that. So let's talk about how that all came to be. So early on, John Tyndall was the first to discover what are known today as bacterial endospores. These are heat-resistant microbial structures. These are structures that bacteria make that are so heat resistant that, just standard boiling will not kill these structures. We're going to learn about bacterial endospores, but it was really John Tyndall who suggested that these endospore structures exist. And it shows how important it is in the field of medicine to make our instruments sterile, completely free of all forms of life, including endospores and viral particles. So in the field of microbiology or in medicine, when we say sterile, that is the gold standard of, free from anything that can cause disease, right? So if a doctor says that an instrument is sterile, this means that it has been autoclaved. We're going to talk about what that device is, but that autoclave destroys every type of bacteria, archaea, protozoa, endospores, viruses, etc., right? Sterile. And before John Tyndall and some of these other early pioneers in medicine came along, this term sterile wasn't at all even a concept, right? It wasn't even thought about to even wash your hands before doing a medical procedure. Now, the development of aseptic techniques really started with some other pioneers here. Robert Koch, another rock star in the field of microbiology. Robert Koch was the first to link a specific microorganism with a specific disease by developing his 4 postulates. These postulates help to verify germ theory. Germ theory stated that germs cause disease, and people didn't buy it. People didn't believe it until Koch linked a particular microbe to a particular disease. Isn't that neat? So for instance, it was Koch who said, it's this bacteria that causes this disease, and I can show it, and I can show it with my 4 postulates. If you follow my postulates in order, then you can link a bacteria to a disease. He used it to link, you guys ever heard of the disease called anthrax poisoning, anthrax? Well, he was the one who showed, using his 4 postulates, that it's the bacterium Bacillus anthracis that causes anthrax. And how did he link the causative agent Bacillus anthracis to the disease anthrax? By using his 4 postulates. Isn't that fascinating? Isn't that neat? And Dr. Oliver Wendell Holmes, he played a role early on. He observed something counterintuitive. Think about this. This doesn't make any sense. Look at this. Dr. Oliver Wendell Holmes observed that mothers who gave birth at home experienced fewer infections, better outcomes, okay, than mothers who gave birth in the hospital. Does that make sense? Why would you have worse outcomes at the hospital than if you give birth at home? Today, it's the opposite, isn't it? Today, you have better outcomes if you have the birth in the hospital, because if there's a complication, there's a medical staff there to help try to fix the issue. But back then, it was a mystery. Now we know why, right? Let me give you some foreshadowing. Well, if you don't know to wash your hands, if you don't know to keep a sterile environment or a disinfected environment, well, yeah, guess what? At hospitals, there are sick people spreading germs. And, if the doctors aren't washing their hands, if no one's washing anything, well, yeah, of course, if you give birth in that kind of environment, your baby may catch some pretty bad diseases, right? And become ill. Next, Dr. Ignaz Semmelweis, who was a pioneer. He had a tragic outcome at the end of his life, but he really was a very famous doctor who showed that the importance of hand washing. He showed that women became infected in the maternity ward after examinations by physicians who had been working in the autopsy room without washing their hands. Can you imagine? Back then, you didn't have, specialties like the mortician or the coroner, touching dead bodies. you could have a doctor who did it all, right? The doctor would go and do the autopsy with a dead body, and then with those germ-ridden hands, run over to help deliver a child, with an expectant mother, in the maternity ward. And then they wondered why, these women became infected, their children, the babies became infected. And so Ignaz Semmelweis was the first to really promote hand washing and say, listen, let's wash our hands before we deliver that baby, especially if we just ran over from the autopsy. And when doing so, he showed that the outcomes, went through the roof, and they improved so much, right? So thank you, Ignaz Semmelweis, for suggesting hand washing. And now that's just commonplace, right? Can you imagine your doctor not washing their hands? But thanks to Ignaz Semmelweis that they do. Then it spread to surgeons, right? Joseph Lister, Dr. Joseph Lister was a surgeon who, would perform surgeries, of course. And he was the first to utilize hand washing and misting operation rooms with antiseptic chemicals. He also treated his surgical instruments with disinfectants and sterilants such as phenol, right? He actually treated his scalpel with phenol, a potent disinfectant. And by doing so, he was able to improve patient outcomes, right? Imagine your doctor not sterilizing the scalpel before cutting you open, right? This Lister was the first surgeon to do so. Now, for the last part of chapter 1, let's talk about how we name, classify, and identify microorganisms. Have you heard of this term nomenclature? Nome meaning name, and clature being kind of classification. So scientists want to give scientific names to organisms. You probably know the scientific name for humans. Can you beat Wicket to answering what is the scientific name for a human? That's right, Wicket. It's Homo sapiens, right? So Homo sapiens is the scientific name for human. And we're going to discover what that scientific name means. Why is it a 2-word epithet? What's the first word represent? What's the second word represent? Do you have to write it a certain way? All that stuff. And we're going to talk about how it relates to microorganisms as well. So again, nomenclature, the assignment of scientific names to various taxonomic categories and individual organisms. Taxonomy is the field of biology that classifies and categorizes organisms based on their relatedness. Classification attempts the orderly arrangement of organisms into a hierarchy of taxa. Do you guys remember the hierarchy of taxa? This is the hierarchy of classification. Domain, that's the most broad, isn't it? The most broad category. How many domains were there? Can you beat Wicket? How many domains are there? That's right, Wicket, as always, 3 domains of life. So if you're an organism, you belong to one of the 3 domains of life. Now, if you're in that domain, you could be subcategorized into a kingdom. So for example, let me give you an example. If we're talking about the domain Eukarya, well, the kingdom could be Animalia, or the animals, or the kingdom could be Plantae, which are the plants, or the kingdom could be fungi, the fungi, right? So you see what I'm saying? You have domains, and under the domain, you can have various kingdoms. Under the kingdoms, you can have various phyla. Under phyla, you can have various classes. And so you're getting more and more and more specific. By the time you get to genus, you're talking about just a handful of very, very closely related organisms. And species is your particular species. A species is a population of individuals that can mate and have fertile offspring. So they can reproduce and have fertile offspring. Okay, so domain is the broad end of this taxonomic hierarchy, species is the most specific end. And so I ask my students to remember this pyramid, remember this taxonomic hierarchy, all the way from domain to species. And the way I remember it, I have a mnemonic device. And here it is. Ready? To remember domain, kingdom, phylum, class, order, family, genus, species, in that order, all I do is I think, dude, King Philip came over for good spices. That's a silly mnemonic device, I know, but it helps me to remember this taxonomic hierarchy in order from domain all the way to species just by remembering that, dude, King Philip came over for good spices, right? So I know it's silly, but it's a good way of remembering this. And the reason I bring this up is because when we're talking about microorganisms, I might say, well, this is the family Enterobacteriaceae, or this is the genus Streptococcus, or this is the species such and such, right? So when I'm talking about a kingdom, you kind of know where in the taxonomic hierarchy I'm at. Or if I'm talking about a phylum, you know what I'm talking about, right? Because these terms come up in microbiology, so we should know how this works. Now, why is it a two-word epithet, right? Homo sapiens, right? Why is there two words? Well, those two words are called the binomial nomenclature. Binomial nomenclature. Bi meaning two, nomial meaning name, the two name nomenclature. It's a combination of the genus name and the species name. So for instance, when we're saying Homo sapiens, the first word is the genus, the second word is the species. In bacteria, you might hear something like this. Look at the bottom here. Staphylococcus aureus. Staphylococcus is the genus. Aureus is the species. Exactly. So anytime you hear these two-word scientific names, now you know what we're talking about. We're talking about binomial nomenclature, the two-word epithet. The first word is the genus. The second word is the species. And it can be abbreviated. Oftentimes in microbiology, these terms, the first term is abbreviated. So for instance, Staphylococcus aureus would be abbreviated as S. aureus. Okay. Now, I want to tell you the rules. You ready for the rules? Both names, the genus and the species, should be italicized if printed on a computer, or a typewriter or something. If it's printed, it needs to be italicized or underlined if written by hand. You need to italicize or underline scientific names, period. You have to do it. Also, the genus name is always capitalized and the species name is lowercase. So you see, Staphylococcus needs to be capitalized, aureus needs to be lowercase. Okay, so these are the rules of the binomial nomenclature. And we're going to be looking at so many different organisms. You've heard of E. coli, right? Well, what does the E stand for? Well, that's its genus, Escherichia. Escherichia coli. I know it's a tongue twister, but that's what it is. Escherichia coli is shortened to E. coli. And of course, I'm going to either italicize that or I'm going to underline it. And I'm going to capitalize Escherichia or E, and I'm going to have a lowercase for coli, for C. And again, I told you what taxonomy is. It's the science of classifying biological species, developed by Carl Linnaeus in the 1700s. This is where we get domain, kingdom, phylum, class, etc., etc. Now, last concept of the chapter. Are you ready for this? Again, when we're looking at the three domains of life, remember the three domains of life are bacteria, archaea, and eukarya. This here, what you're looking at, is known as the phylogenetic tree. This means a tree of life showing different organisms and how they're related to one another. All of us share a common ancestor, right? Remember, we all share a common ancestor. And that is shown by this one line here at the bottom. Now, when there's this node and you see how it diverges from this node into two branches, that dot there, here, this spot is where there was a divergence, right? This is a common ancestor where there was a divergence. Now, that divergence led to the bacteria and the archaea eukarya. Okay. Now, look at this. What does this phylogenetic tree suggest to you? Look, there was a common ancestor for all living organisms. Then there was the last common ancestor. There was a branch off to bacteria, and the other branch became the archaea and the eukarya. So it looks like eukarya and archaea, look at this, eukarya and archaea share a more common ancestor than either does to bacteria. Do you know what this means? This means because of this right here, you see what I'm circling right here, because archaea and eukarya are known to share a more common ancestor than they do with bacteria, archaea and eukarya are technically more closely related to one another than either is to bacteria. This should blow your minds, right? This should blow your mind because remember, I said bacteria are prokaryotic, archaea are prokaryotic. So you would think they're very closely related, right? But actually, no, they're not. They're actually quite distantly related. If you look at them genetically, archaea are way more closely related to us eukarya than they are to bacteria. Isn't that interesting? So if I ask you on the exam, are archaea more closely related to bacteria or to eukarya, what would you say? Can you beat Wicket? That's right, Wicket. Archaea are more closely related to eukarya than either is to bacteria. Isn't that interesting and fascinating? And thanks to genetics for making that discovery. Before, archaea and bacteria used to be grouped together in taxonomy, but after they discovered how genetically different archaea is from bacteria, they belong in completely different domains. Isn't that neat? So very fascinating stuff. I hope this was a good review of Chapter 1. Welcome to Biology 2420. I want to welcome you. I hope you enjoyed this lecture series, and I can't wait to bring you more.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.